Hi everyone. This is an introduction to MNase-seq. MNase-seq, which stands for micrococcal nuclease digestion with deep sequencing, is a method to identify open chromatin in DNA. To begin with, we must first understand some basics about chromatin structure. Eukaryotic DNA can be represented by the beads on a string model, where each bead corresponds to the DNA double helix wrapped around a core of 8 histone proteins forming a nucleosome. This aids in the condensation of chromatin, which is further compacted into chromatin fiber and eventually into a metaphase chromosome. This type of chromosome representation is what you probably think about when imagining chromosomal DNA. But chromatin is not always that compacted, and in fact is much more open during interphase. There are two states of chromatin, open and closed. Open chromatin is accessible to nucleases such as MNase, whereas closed chromatin is not. Open chromatin is again DNA that has few or no histone proteins and is therefore accessible, while closed chromatin is DNA that is tightly compacted into nucleosomes and is inaccessible. There are functional consequences to different chromatin states. The DNA that is in areas of open chromatin is capable of being transcribed, whereas DNA in areas of closed chromatin is unable to be transcribed by RNA polymerase. So compacted genes are usually not expressed. Through changes in the location and density of nucleosomes, certain genes are regulated by allowing or not allowing transcription to occur. Now that we understand the concepts of open and closed chromatin, we can look at the processes of MNase-seq. To begin, cell nuclei must be isolated from the cells of interest. These nuclei are then treated with micrococcal nuclease, an enzyme derived from the bacteria Staphylococcus aureus. This enzyme is a non-sequence-specific endo-exonuclease that can cleave the DNA in areas where no protein is bound, i.e. the areas of open chromatin that were mentioned earlier. Both strands of exposed DNA get cleaved by MNase in the presence of calcium ions. This leaves short sections of DNA still bound to transcription factors or histone proteins intact. The fragmented DNA can then be purified to isolate DNA. The sections of nucleosome-protected DNA are typically 150 base pairs in length, and DNA fragments bound to transcription factors are generally much shorter than this. Then the sections of DNA bound to histone proteins or transcription factors are isolated, the proteins are stripped away, the DNA ends are repaired, adapters are ligated, and sequencing is performed using one of the next generation short-read sequencing platforms such as Illumina. We can then map the DNA fragments to the genomic DNA sequence to determine the locations of nucleosomes and transcription factors at a whole genome level. This information can be cross-referenced with RNA-seq data obtained from the same tissues to correlate chromatin accessibility profiles with sites of active transcription. Recently, MNase has been leveraged in novel sequencing methods. The cut-and-run method combines MNase with chromatin immunoprecipitation to specifically isolate and sequence DNA bound to nuclear proteins, such as transcription factors. A second novel MNase-seq method is single-cell MNase-seq. This method is very similar to MNase-seq, but it takes advantage of fluorescent activated cell sorting to isolate single cells that are then treated with MNase and their DNA is adapted, PCR amplified, and then sequenced. MNase-seq is one of several chromatin accessibility assays available today along with DNA-seq, FAIR-seq, and ATAC-seq. These methods all are very useful techniques in the researcher's toolbox for understanding chromatin structure, identifying regions of open versus closed chromatin, and pinpointing nucleosome and other binding sites. With that, I hope you gained a brief understanding of MNase-seq.